Welcome to Advanced Exercise Physiology. This is Chapter 10, looking at respiration during exercise. The objectives of this chapter are as follows. 1. To explain the principal function of the pulmonary system. 2. To outline the major anatomical components of the respiratory system. 3. To list major muscles involved in inspiration and expiration at rest and during exercise. 4. To discuss the importance of matching blood flow to the alveolar ventilation in the lung. 5. To explain how gases are transported across the blood-gas interface in the lung. 6. To discuss the major transportation modes of oxygen and carbon dioxide in the blood. 7. To discuss the effects of increasing temperature, decreasing pH, and increasing levels of 2,3 dpg on the oxygen hemoglobin dissociation curve. 8. To describe the ventilatory response to constant load, steady state exercise. What happens to ventilation if exercise is prolonged and performed in a high temperature humid environment? 9. To describe the ventilatory response to incremental exercise, and to look at what factors are thought to be contributing to the linear rise in ventilation at work above 50% to 70% of VO2 max. 10. To identify the location and function of chemoreceptors and mechanoreceptors that are thought to play a role in the regulation of breathing. And 11. To discuss the Humoral theory of respiratory control during exercise. For those of you taking notes, here is an outline focusing on the major concepts focused in Chapter 10. Starting with an introduction, we have pulmonary respiration, which involves ventilation and the exchange of oxygen and carbon dioxide in the lungs. There's also a cellular respiration, where oxygen utilization and carbon dioxide production takes place in the tissues. The purpose of the respiratory system during exercise is gas exchange between the environment and the body and regulation of the acid-base balance during exercise. If we look at the function of the lung, we see the means of gas exchange between the external environment and the body, which is replacing oxygen, removing carbon dioxide, and a regulation of acid-base balance. We see that there's ventilation, which is the mechanical process of moving air into and out of the lungs, and also diffusion takes place, which is the random movement of molecules from an area of high concentration to an area of low concentration in the lungs. In summary, the primary function of the pulmonary system is to provide a means of gas exchange between the environment and the body. In addition, the respiratory system plays an important role in the regulation of the acid-base balance during exercise. Moving on to the structure of the respiratory system, we see that it is made up of organs such as the nose and nasal cavities, the pharynx and larynx, the trachea and bronchial tree, and the lungs, which contain the alveoli, which is the site of gas exchange. In addition, there's the diaphragm, which is the major muscles used in inspiration. Looking more closely at the lungs, we see that they are enclosed by membranes called pleura. There's a visceral pleura, which is on the outer surface of the lung. There's also a parietal pleura, which lines the thoracic wall, and there's an interpleural space. The interpleural pressure is lower than the atmosphere, which prevents collapse of the alveoli. Now you can see in the diagram all the major organs of the respiratory system which we just described. In addition, this diagram shows the positions of the lungs, diaphragm, and pleura. Now looking at conducting and respiratory zones, we'll see that the conducting zone is where we conduct air to the respiratory zone. It humidifies, warms, and filters the air. The components of the conducting zone are the trachea, bronchial tree, and bronchioles. If we move to the respiratory zone, we see that this is where the exchange of gases between the air and the blood happens. The components are the respiratory bronchioles and the alveolar sacs, where surfactant prevents the alveolar collapse. Here is a diagram displaying the conducting and respiratory zones and how they connect. In addition, here's a more detailed diagram of the bronchial tree. We also have a diagram of the type 2 alveolar cells. Again, all of these diagrams are outlined in your book in Chapter 10. In summary, anatomically, the pulmonary system consists of a group of passages that filter air and transport it into the lungs, where gas exchange occurs within teeny air sacs called alveoli. Now, looking at the mechanics of breathing, we see that the movement of air occurs via bulk flow. There's movement of molecules due to the pressure difference. We have inspiration, where the diaphragm pushes downwards and the ribs lift outward. This allows the volume of the lungs to increase, and the interpulmonary pressure is lowered. Conversely, during expiration, the diaphragm actually relaxes and the ribs are pulled downward. As a result, the volume of the lungs decreases and the interpulmonary pressure is raised. Here's another diagram displaying the mechanics of inspiration and expiration. In addition, this diagram shows us the muscles that are involved of respiration. Now, talking a little bit more about the respiratory muscles and exercise, we want to ask the question, do respiratory muscles fatigue during exercise? Historically, it is believed that respiratory muscles do not fatigue during exercise. However, current evidence suggests that respiratory muscles in fact do fatigue during exercise. This exercise would be prolonged greater than 2 hours or at high intensities, approximately 90 to 100% of an individual's VO2 max. The next question is, do respiratory muscles actually adapt to training? And the answer is emphatically yes. We see an increased oxidative capacity, which improves respiratory muscle endurance, and a reduced work of breathing. Now, if we take a look at airway resistance, we see that airflow depends on the pressure difference between the two ends of the airway and the resistance of the airways. We also see that airway resistance depends on diameter. This is evident in chronic obstructive lung disease and asthma-induced during exercise. Looking more closely at exercise-induced asthma, we see that the asthma results in a bronchospasm. This is narrowing of the airways, which leads to an increased work of breathing and shortness of breath. There are many potential causes of this asthma. Now, looking at exercise-induced asthma, we see that the bronchospasm is during or immediately following exercise. This may actually impair exercise performance. If we look at exercise and chronic obstructive lung disease, we see that during chronic obstructive lung disease, there's an increased airway resistance due to a constant airway narrowing, which leads to a decreased expiratory airflow. This includes two different types of lung diseases. This would be chronic bronchitis, where excessive mucus blocks the airways, and emphysema, where airway collapse leads to an increased resistance. We ultimately see an increased work of breathing, which leads to shortness of breath and may interfere with exercise and activities of daily living. In summary, the major muscles of inspiration is the diaphragm. Air enters the pulmonary system due to the interpulmonary pressure being reduced below the atmospheric pressure in bulk flow. At rest, expiration is passive. However, during exercise, expiration becomes active using muscles located in the abdominal wall. For example, the rectus abdominis and the internal obliques. The primary factor that contributes to airflow resistance in the pulmonary system is the diameter of the airway. Now, if we look at pulmonary ventilation, we see that the amount of air moved in or out of the lungs per minute is the volume. We have a tidal volume, which is the amount of air moved per breath. We have a breathing frequency, which is the number of breaths per minute. We have an alveolar ventilation, which is the volume of air that reaches the respiratory zone. And we have dead space ventilation, which is the volume of air remaining in the conducting airways. In summary, pulmonary ventilation refers to the amount of gas moved into and out of the lungs. The amount of gas moved per minute is the product of tidal volume times breathing frequency. Now, if we look at pulmonary volumes and capacities, the first thing we would look at is vital capacity, which is the maximum amount of gas that can be expired after a maximum inspiration. There's also a residual volume, which is the volume of gas remaining in the lungs after maximum expiration. And we have a total lung capacity, which is the amount of gas in the lungs after a maximum inspiration. The chart and table below lists the definitions of pulmonary volumes and capacities. Please reference this table in your book. The following diagram shows the different lung volumes and capacities that we just discussed. Now, if we look at spirometry, we see that it's the measurement of the pulmonary volumes and the rate of expired airflow. It's useful for diagnosing lung disease such as chronic obstructive lung disease. The spirometric tests include vital capacity, again, which is the maximal volume of air that can be expired after a maximal inspiration, a forced expiratory volume, which is the volume of air expired in 1 second during a maximal expiration, as well as the forced expiratory volume over the vital capacity ratio, which approximately greater than or equal to 80% is normal. This is simply a diagram of a computerized spirometer, which can be used for diagnosis. And in this diagram below, we see the forced expiratory airflow used to diagnose airway obstruction. In summary, the pulmonary volumes can be measured using spirometry. The vital capacity is the maximum amount of gas that can be expired after maximal inspiration. Finally, residual volume is the amount of gas left in the lungs after a maximal expiration. Now, if we look at the partial pressure of gases, we would reference Dalton's law, which is that the total pressure of a gas mixture is equal to the sum of the pressure that each gas would exert independently. Below, you'll see a variety of calculations of the partial pressure for the gases of oxygen, carbon dioxide, and nitrogen within the air. Now, looking at the diffusion of gases, we would reference Fick's law of diffusion, which is the rate of gas transfer is proportional to the tissue area, the diffusion coefficient of the gas, and the difference in the partial pressure of the gas on the two sides of the tissue, and inversely proportional to the thickness. In this diagram, we can see an example of the partial pressures of oxygen and carbon dioxide and gas exchange. In summary, gas moves across the blood-gas interface in the lung due to simple diffusion. The rate of diffusion is described by Fick's law, and diffusion is greater when the surface area is large and the driving pressure is high. Moving on to blood flow to the lung, we again reference the pulmonary circuit. It has the same rate of flow as the systemic circuit, but it is at lower pressure. When standing, most of the blood flow is to the base of the lungs due to the gravitational force. However, during exercise, more blood flow is to the apex of the lung. This is again a reference diagram to the pulmonary and systemic circulation. And this is regional blood flow within the lung. In summary, the pulmonary circulation is a low-pressure system with a rate of blood flow equal to that of the systemic circuit. In a standing position, however, most of the blood flow to the lung is distributed to the base of the lung due to gravitational force. Now, if we look at the ventilation-perfusion relationships, we can first examine the ventilation-perfusion ratio. It indicates the matching of blood flow to ventilation. Ideally, the ratio would be 1.0. At the apex of the lung, it is underperfused, however, and the ratio is less than 1. And at the base of the lung, it is overperfused, where we have a ratio greater than 1. However, if we look at it during exercise, we see that light exercise improves the V over Q ratio, where heavy exercise results in a V over Q ratio of inequality. The following diagram represents the ventilation-perfusion ratios that we just discussed. In summary, efficient gas exchange between the blood and the lung requires proper matching of blood flow to ventilation, called the ventilation-perfusion relationships. The ideal relationship of ventilation to perfusion is 1.0 or slightly greater, since this ratio implies perfect matching of the blood flow to ventilation. Now, if we look at the oxygen transport in the blood, we see that 99% of oxygen is transported bound to hemoglobin. We have oxyhemoglobin, where hemoglobin is bound to the oxygen, and deoxyhemoglobin, where the hemoglobin is not bound to the oxygen. We see that the amount of oxygen that can be transported per unit volume of blood is dependent upon the hemoglobin concentration, where each gram of hemoglobin can actually transport 1.3 milliliters of oxygen. Now, if we look at the oxygen content of the blood, at 100% hemoglobin saturation, we see that males can carry 200 milliliters of oxygen per liter of blood, while females can only carry 174 milliliters of oxygen per liter of blood. Now, if we examine the oxyhemoglobin dissociation curve, we see that deoxyhemoglobin plus oxygen equals oxyhemoglobin. Now, the direction of this reaction depends on the PO2 of the blood and the affinity of hemoglobin and oxygen. At the lung, we have a high PO2, which leads to the formation of oxyhemoglobin. However, at the tissues, we have a low PO2, which leads to the release of oxygen into the tissues. This represents the oxygen-hemoglobin dissociation curve that we just discussed. Now, if we examine the effect of pH, temperature, and 2,3 dpg on the oxygen-hemoglobin dissociation curve, we see that pH decreases and lowers the hemoglobin-oxygen affinity. This results in a rightward shift of the curve and favors offloading of the oxygen at the tissues. If we look at temperature, we see that increased blood temperature lowers the hemoglobin-oxygen affinity, and this as well leads in a rightward shift of the curve. Finally, looking at 2,3 dpg, we see that this is the byproduct of red blood cell glycolysis. It may result in a rightward shift of the curve as well. We see this during altitude exposure, but is not a major cause of the rightward shift during exercise. Here's a diagram that shows the effect of pH on the oxygen-hemoglobin dissociation curve, where the decrease in pH leads to a rightward shift. Here as well is a diagram of the effect of temperature on the oxygen hemoglobin dissociation curve, where we can see that the increase in temperature leads to a rightward shift. Now, if we again look at the oxygen transport in the muscle, we see that myoglobin actually shuttles the O2 from the cell membrane to the mitochondria. We know that myoglobin has a higher affinity for oxygen than the hemoglobin. This happens even at a low pressure of oxygen and allows the myoglobin to store the oxygen, which leads to an oxygen reserve within the muscles. Here we see the dissociation curve for myoglobin and hemoglobin. Now, if we look at the carbon dioxide transport in the blood, we see that 10% of it is dissolved in the plasma, 20% is bound to hemoglobin, and 70% is in the form of bicarbonate. In the tissue, we see that the hydrogen binds to the hemoglobin, which allows the bicarbonate to diffuse out of the red blood cell into the plasma. And we also see the chloride diffuses into the red blood cell, which is called the chloride shift. At the lung level, we see that the oxygen binds to the hemoglobin and dries off the hydrogen, and the reaction reverses to release the carbon dioxide. Here is a diagram of the carbon dioxide transport in the blood at the tissue. And here is a diagram of the carbon dioxide transport in the blood at the lung. In summary, more than 99% of the oxygen transported in the blood is chemically bound to the hemoglobin. The effect of the partial pressure of oxygen on the combination of the oxygen with hemoglobin is illustrated by the S-shaped oxygen hemoglobin dissociation curve. An increase in body temperature and a reduction in the blood pH results in a right shift of the oxygen hemoglobin dissociation curve and a reduced affinity for hemoglobin and a reduced affinity of hemoglobin for oxygen. In addition, carbon dioxide is transported in the blood in three forms. One, dissolved carbon dioxide, which 10% of carbon dioxide is transported this way. There's also carbon dioxide bound to hemoglobin, and about 20% of the blood carbon dioxide is transported via this form. And we also have bicarbonate, where 70% of the carbon dioxide found in the blood is transported as bicarbonate. Now, if we look at ventilation acid-base balance, we see that pulmonary ventilation removes hydrogen from the blood by the bicarbonate reaction. This results in an increased ventilation in the carbon dioxide exhalation, and it reduces the partial pressure of carbon dioxide and the hydrogen concentration, or the pH, with a pH increase. Now, decreased ventilation results in a buildup of carbon dioxide, where we see an increased partial pressure of carbon dioxide and hydrogen concentration, which leads to a pH decrease. In summary, an increase in pulmonary ventilation causes exhalation of additional carbon dioxide, which results in a reduction of blood partial pressure of carbon dioxide and a lowering of the hydrogen ion concentration, which increases the pH. Now, if we start to look at rest to work transitions, we see that at the onset of a constant load at submaximal exercise, there's an initial increase in the ventilation rapidly, and then there's a slower rise towards the steady state. The partial pressure of oxygen and the partial pressure of carbon dioxide are relatively unchanged. We do see a slight decrease in the partial pressure of oxygen and an increase in the partial pressure of carbon dioxide, however. This is a diagram showing the transition from rest to exercise for the partial pressure of oxygen, carbon dioxide. If we look at prolonged exercise in a hot environment, we see that during prolonged submaximal exercise in the hot humid environment, the ventilation tends to drift upward. we see that this leads to an increased blood temperature, which affects the respiratory control center. However, there is little change in the partial pressure of carbon dioxide, and we see the higher ventilation not due to the increase in the partial pressure of carbon dioxide. This following diagram represents how exercise is influenced in a hot and humid environment like we just discussed. Look at the incremental exercise of an untrained subject. We see that in ventilation there's a linear increase up to approximately 50 to 75% of the VO2 max. Beyond this point, we see an exponential increase. In addition, the subject will hit the ventilatory threshold, which is the inflection point where the volume of expiration increases exponentially. We also see in the partial pressure of oxygen that it is maintained within a very close range of the resting value. Now, if we look at the incremental exercise in an elite athlete, we see that in ventilation, the ventilatory threshold occurs at a higher percentage of VO2 max. We also see that there's a decrease in the partial pressure of oxygen at near maximal work. We call this hypoxemia. This is due to the ventilation-perfusion ratio mismatch, and we also see a shorter red blood cell transit time in pulmonary capillaries due to a high cardiac output. The two charts below represent the ventilatory response to incremental exercise in untrained and elite athletes. In summary, at the onset of constant load submaximal exercise, ventilation increases rapidly, followed by a slower rise toward a steady state value. Arterial partial pressure of oxygen and carbon dioxide are maintained relatively constant during this type of exercise. During prolonged exercise in hot humid environment, ventilation drifts upward due to the influence of rising blood temperature on the respiratory control center. We see that incremental exercise results in a linear increase of the ventilation up to approximately 50 to 70% of the VO2 max. At higher work rates, ventilation begins to rise exponentially. This ventilatory inflection point has been called the ventilatory threshold. Now, if we look at the control of ventilation at rest and focus on inspiration and expiration, we see that it is produced by contraction and relaxation of the diaphragm. We also see that it is controlled by somatic motor neurons in the spinal cord. It is controlled by the respiratory control center in the medulla oblongata. Focusing on this respiratory control center, we see that stimulus for inspiration comes from 4 respiratory rhythm centers. Some are in the medulla, and others are in the pons. Now, if we look at the group pacemaker hypothesis, this suggests that the regulation of breathing is under redundant control. Now, if we look at the input to the respiratory control center, we see that this comes from humoral chemoreceptors. We have central chemoreceptors, some that are located in the medulla, and they look at the partial pressure of carbon dioxide and hydrogen concentrations in the cerebral spinal fluid. We also have peripheral chemoreceptors, which are located in the aortic and carotid bodies and look at the partial pressure of oxygen, carbon dioxide, and also hydrogen and potassium in the blood. There's also neural input that comes from the motor cortex and skeletal muscle mechanoreceptors. Here's a diagram that represents the brainstem and the respiratory control centers. And this is a diagram that shows the location of the peripheral chemoreceptors. Next, we have a diagram that shows the effects of the arterial partial pressure of carbon dioxide on ventilation, where we see an increase in arterial partial pressure of carbon dioxide leads to an increase in ventilation. Next, we see the effect of arterial partial pressure of oxygen on ventilation. Now, ventilatory control during exercise. At submaximal exercise, we see that there's a primary drive. This comes from the higher brain centers, or the central command. It is fine-tuned by humoral chemoreceptors and neural feedback from muscles. However, at heavy exercise, we see an alinear rise in the ventilation. This is due to an increased blood concentration of hydrogen from lactic acid, which stimulates the carotid bodies. Also, in potassium, body temperature, and blood catecholamines might contribute to this. The following chart is a summary of the respiratory control during submaximal exercise. Now, if we see how training reduces the ventilatory response to exercise, we see that there's no effect actually on the lung structure. But ventilation is lower during exercise following training. We see that exercise ventilation is about 20 to 30% lower at the same submaximal work rate. If we look at the mechanism, we see changes in the aerobic capacity in the locomotor muscles. There is a result in less production of hydrogen and less afferent feedback from the muscles to stimulate breathing. This chart below shows the effects of endurance training on ventilation during exercise, where after training we have a decrease in ventilation. In summary, current evidence suggests that the normal rhythm of breathing is generated by the interaction between 4 separate respiratory rhythm centers located in the medulla oblongata and the pons. At rest, the breathing rhythm is dominated by the pacemaker neurons in the pre-Bonzinger complex. During exercise, however, the pre-Bonzinger complex interacts with the retrotrapezoidal nucleus along with 2 additional regulatory centers in the pons to regulate breathing. The coupling of these respiratory control centers to regulate breathing involves both positive and negative feedback to achieve tight control. We also see that input into the respiratory control center to increase ventilation can come from both neural and chemoreceptor sources. Neural input may come from a higher brain center, or it may arise from receptors in the exercising muscles. The humoral chemoreceptor input may arise from central chemoreceptors and peripheral chemoreceptors. The central chemoreceptors are sensitive to increases in the partial pressure of carbon dioxide and decreases in pH. The peripheral chemoreceptors in the carotid arteries are the most important and are sensitive to increases in the partial pressure of carbon dioxide and increases in the partial pressure of oxygen or pH. And finally, the primary drive to increase ventilation during exercise comes from the higher brain centers or the central command. Also, humoral chemoreceptors and neural feedback from working muscles act to fine-tune the ventilation. Now, looking at the effect of training on ventilation, we see again that there's no effect on lung structure and function at rest. Normal lung exceeds demand for the gas exchange, and adaptation is not required for the lung to maintain the blood-gas homeostasis. One exception is elite endurance athletes. There is a failure of the lung to adapt to training, which results in hypoxemia. Now, if we ask the question, does the pulmonary system limit exercise performance? We see that at low to moderate intensity exercise, the pulmonary system in fact does not act as a limiting factor. Now, if we look at maximal exercise, however, historically it was not thought to be a limitation in healthy individuals at sea level. However, new evidence suggests that respiratory muscle fatigue does occur during high-intensity exercises greater than 90% of an individual's VO2 max. In addition, there may be a limiting in elite endurance athletes where 40 to 50% might experience hypoxemia. In summary, the pulmonary system does not limit exercise performance in healthy young subjects during prolonged submaximal exercise where the work rates are less than 90% of the VO2 max. In contrast to submaximal exercise, new evidence indicates that the respiratory system might experience respiratory muscle fatigue and may be a limiting factor in exercise performance at work rates greater than 90% of the VO2 max. Further, incomplete pulmonary gas exchange may occur in some elite athletes and limit exercise performance at high exercise intensities. That concludes the content for Chapter 10. I'd like to provide some study questions that will further challenge you to understand the material and make sure that you have a grasp of what we've covered. Question 1. What is the primary function of the pulmonary system, and what secondary function does it serve? 2. List and discuss the major anatomical components of the respiratory system. 3. What muscle groups are involved in the ventilation during rest and during exercise? 4. What is the functional significance of the ventilation-perfusion ratio? Also, how would a high VQ ratio affect gas exchange in the lung? 5. Discuss those factors that influence the rate of diffusion across the blood-gas interface in the lung. 6. Graph the relationship between the hemoglobin oxygen saturation and the partial pressure of oxygen in the blood. What is the functional significance of the shape of the O2 hemoglobin dissociation curve? Also, what factors affect the shape of the curve? 7. Discuss the modes of transportation for carbon dioxide in the blood. 8. Graph the ventilatory response in the transition form to rest to constant load submaximal exercise. What happens to ventilation if the exercise is prolonged and performed in a hot, humid environment, and why? 9. Graph the ventilatory response to incremental exercise and label the ventilatory threshold. What factors might explain the ventilatory threshold? 10. List and identify the functions of the chemoreceptors that contribute to the control of breathing. 11. What neural afferents might also contribute to the regulation of ventilation during exercise? And 12. Discuss the control of ventilation during exercise. This concludes Chapter 10, Respiration During Exercise. Please reference your text or review this lecture if you have any questions, or feel free to email me at any point. Thanks.